we said if you start with the matrix that we wrote really last week, is this is transition matrix. So what is the rate at which you go from here to here? You have 0 molecules that are bound, and you have total m molecules. So what is the rate of having one molecule bound? You need to take the rate of binding, which is lambda, the number of total number of possibilities m, and the total number of free binding sites is s. There is no nothing bound, right? And so you can write the rates like this. So this was one.
M is fixed here, or is M decreasing?
Yeah. 1 sec. And so the second direction of the matrix is that when m decreases, and so now we enter into the part of the chain where now you can have 0 bound and 1 particle disappeared, and this happened with a rate which is exactly given by the Poissonian rate, which is mu multiplied by the total number of available molecule, which is m. All right, and we can continue now. Suppose in this line, no molecule disappeared, and we just get binding. And we arrive to here with T and M -1, correct? And again, once you're here, you can calculate the rate of transition from here to any of these. And up to where are we going? We need to go up to so if we have m molecules, so this is a state that interests us. We have T-bound molecule, T-bound molecules. Now, when is this going to fail? If the number of bound molecules, I mean, if there is not enough molecule to reach the threshold T, then wherever we continue, we'll never reach, right? So let me write down the matrix. So we have then 0, and then we end up here in 0 and T. And so we can have here, so the number of molecules has decreased up to the threshold. So which means that below this threshold, I will never have enough molecule to bind. So then this goes to, now we have binding. So we have 1 and T, and so on. And then we arrive to here, to the total number of bound molecule, which is going to be T, when we are left with exactly T. Right? And so what is the ensemble of state we're interested in is going to be these ensembles. So we start, this is a starting point. We start here. And now we let evolve the process. So now how do you end up here, here, or here? So how you end up here, this is not very hard. You just start here, you have no jumps, just continue this way. How do you end up here? This is more complicated because what can happen? If you want to end up in here, there are many ways. You have to count all the paths that can go from here to here. So remember, there is no way when you're bound to come back, and when you disappeared, you cannot appear again. You are dead forever. So for example, how you end up in here? You end up in here by any choice. You go from here to a certain point. Then you make a jump here, and then you continue this way. So how many possibilities there are? As many possibilities as there are to come once from here to here, which is exactly t plus 1, right? And so now when you are 1 and 2, then you have to count all the pairs at which you can jump in here for the first time, then you wait and you jump for the second time. So this you can write down. So I don't know if we wrote down last week the entire Markov chain.
If we're assuming we're in that final state, Tm or Tm minus 1, are we still evolving the system so we can go from Tm to Tm minus 1 down?
You can continue here, but
I was thinking, because obviously if you start off right at the top, you end up at Tm, are we saying, right, we're stopping there, and we're going to count everything we've got now? Or we're allowing it to evolve?
You're allowed to continue, but what I'm saying is that for the calculation, I just need to calculate the probability of those states. And actually, I am interested in the sum. So what we are interested in, what's the probability? So to answer your question, let's understand what are we looking for. What is the probability of ever reaching the threshold, that is the number of MS molecules when you finish your process? It's all the molecules are not gone. That let's call MS infinity, so those are the molecules that are ultimately bound, equals a threshold T. So this is exactly the probability, this is the sum of the state here, this is the sum of the probability of ending up with T molecule and having exactly M no loss, plus, so let's write it the sum, plus the next one, you have T, and there was M minus 1 up to infinity, and then we go up to PT and T. This is what we are interested in calculating. So obviously you can continue, but there's not going to help to calculate this, because once you are in here, it doesn't contribute. All right, so for example, again, I forgot if I'm, okay, I can write just the middle term for the transition probability, which is P, K, and M. I think we wrote it last week, but let me write it in any case. So last week we looked at if you start at something in the middle here, what was the transition from the guys above on the left, and where things were going, it's the transition from the right or below. So you have to write the transition probability for a two-dimensional Markov chain. And so now we use the fact that you have, in all cases, every transition is Poissonian. So you can, if you start from K minus 1 and M, this means that you have to have one binding. So you start with S0 minus K plus 1, M minus K plus 1, right? So this is the same as in here. What we said that to bind, really it's proportional to the number of free sites multiplied by the number of free binding sites, which is here, for when you start from K minus 1. Then there is the case where you are going to, particle is going to die, M plus 1 minus K, and you start from M plus 1 molecules, and there was no binding, minus the probability that nothing happened, which is S0 minus K, plus mu multiplied by M minus K, P K of M. And this is true, so now we have to be very careful. This is true for T minus 1 between M and M0, and K between 0 and T. And so last week, we noticed, I did it very carefully, I don't want to spend a lot of time on this, we did very carefully what happened when we have, what are the boundary conditions when you reach, when you are imposing a threshold. You remember last week we said when we have ms molecule equals to T, you can start with one bound molecule, then you go to make a transition to 2 and transition to T. T is a threshold. So in principle, as you mentioned, it can continue up to m molecules, or total m, or the minimum of m and s. So all the sites here are filled. Let's say s. Let's suppose s is less than m. But if you impose a threshold, which means that your system stops when you reach the threshold, you have to modify the Markov chain such that this rate that is coming back is not zero. Because when you are in this state, you become absorbing. It becomes an absorbing state. So this k minus 1 becomes 0. And this is how last time we derived the first time to a threshold by putting this rate to 0. So here's the same. Now we have to calculate this, so you have to give other conditions, which I'm not going to write, just this is the Markov chain. Now you can look at the solution of this Markov chain going into infinity, where you know that in the middle, everything will be 0. If you are in this state, this is an absorbing state, so this p infinity here is not 0, right? So either you are here, or you end up in the rest here, which means you didn't reach the threshold. And so you can, again, I'm not writing all this term for the boundaries. But what is important is a normalization, that the sum of tk and m has to be equal to 1 for k equals 0 to n0 and m equals 0 to n0. All right. Okay, so now let me tell you how do you calculate this quantity in a more just with the hand-waving argument, and the calculation can be looked into the paper. So how do you calculate the probability of the first one, for example? What's the probability of this? The probability of this is once you start here, that you made a transition from here to here. So this is the probability. So this is going to be the ratio of the rate here divided by the sum of the two rates, which is the probability of making a transition from here to here. And then you repeat this up to here. So the probability of ending into this so I would like to erase this Markov chain. And you will see, I mean, you can, again, solve all the Markov chains, which is systematically can be done using elementary calculation. But let me give you, so the argument, the intuition is correct. So the PT infinity is simply the product of the binding rate. So lambda s minus I. You make the product from I 0 to t minus 1. And then divided by mu, so this is the rate here, mu. Okay, there is an m that I have cancelled everywhere, msi, sorry, mu plus lambda s minus I plus mu. Right, in principle the rate is, there is an m here in front of everybody. So that's the rate of transition from the first one is lambda ms, but then I moved the m out. Right? So it becomes this. So this is the rate, this is the probability each time. What's the probability? You take the ratio of this rate divided by the sum, which is the probability of making this compared to this one, and you repeat it, you multiply this by this term. Okay, and now how do you get the other ones? The other ones are basically can be derived as this product here, except that somewhere you're making a transition this way. So which means that you have to choose the index at which you switch from the first line to the second one. So this can happen at any of those lines. So you have to sum over all these possibilities. So this will give you the second term. And you do the same for the last one. So ultimately, this probability can be written in terms of this p infinity. This is PTM. PTM. Yeah, PTM 0, okay, whatever m, infinity, 1 plus, and then you get the sums over all possibilities, which is the sum over I1 between 0 and t minus 1. And then you have the transition which come from the mu term. So again, there is an m that will cancel. So this is mu divided by lambda s minus I plus mu. But this is for the first one. And then for the second one, you're going to get here a double sum, which is A1, 0. So of course, once you make a jump here, you cannot go back. So the second index has to be above the first one. So A1 less than A2, and t minus 1, and then you get this mu, and the product for ik of the mu lambda s minus ik plus mu. And until you reach to the last one. All right, and so you end up with something like this, which is very unpractical to calculate, except in some limit where s is much bigger than the threshold, so the I here really do not count. And then, if you don't care about the I, it's really like throwing m particle and looking at the distribution of having t out of m particles that are bound. And then this is simply your binomial distribution. So this is what you can find, is that in some limit where, so I think it was t divided by s, which is large. S is much bigger than the threshold. Then this total, let's call it total p. P is this quantity. P here is simply just the sum of the binomial distribution L and K. When you start, obviously, for at least t molecular bound, you need to have at least t here. And you can have, of course, t plus 1, so whatever. So this is t and m. And then this is the rate, the ratio of this. So the probability is lambda s plus mu. This is binding versus killing to power k. And then this is 1 minus this. So this should be mu divided by lambda s plus, yeah, to power n or m minus k, which is really the Bernoulli distribution. All right, so this is what I wanted to say last time, that this isn't practical to calculate, but at least you can put that on the computer. Last time, what was very nice is we could give explicit expression for this time. You can also calculate another interesting thing, but I don't want to do that now, is now we calculated the probability for this to happen. Now you can calculate the conditional time for this to happen. That is, conditions that you have reached those states, how long it takes. And then, so you have to look again at all the paths. So once you have the probability for one path, you just have to, this is not very hard, you just have to use the time. So the time for this is 1 over the rate. So you just, each time you have a probability, you multiply this probability by the time, and you end up with the mean conditional passage time. All right, so I think I'll stop here for this. Again, this was from last week class. We have many examples where this was relevant. One was for cell division, where we looked at the kinetochore, which is a process when cells have to divide. You have to make sure that the chromosomes are well aligned, and you have a finite number of them. I don't know, depend on the species. Let's say you have 16 or whatever. So you have a finite number, and you want to make sure that they are all attached. I mean, the cell makes sure this, and then they can start dividing. And again, this is a mean time to threshold, because you want to make sure that this threshold is reached before you divide. And we use this approach to estimate rate constant from data. So this was one application. I mentioned the application about synapses at the beginning. And the third application that we had on this process was the work we did with Kevin Barrage, about the probability that a messenger RNA would escape the nucleus protected by microRNAs. So again, you had DNA, messenger RNA. The messenger RNA had some binding sites. So you ask the following question: how long it takes for this to escape through nuclear pore, small hole, here, before it is degraded by a killer, whatever the killer molecule is. So if you have, let's say, all of these molecules bound here, it is destroyed. So no way, no chance to go out. So there was in the game, a molecule that was actually protecting. That is, if the blue molecule is here, the red cannot come, and so it is protected. And so the idea, again, if you have a threshold, for example, if you assume that when the number of red reach a threshold C, that is threshold T, it is destroyed. So now you can have competition of this blue molecule that can come by diffusion and can be removed by detachment. At the same time, you have the red one that can come and leave. So the blue and red compete for the sites. If the red reaches the threshold, the game is over. What's the probability that you reach here before your threshold is reached? This is exactly the type of framework we've developed here. I don't have time to, it will be an additional hour. So what you have to look at here is the joint probability of having the total number of sites and the number of red crosses bound. Assuming that you have enough of them so that when they are released, they are in excess. So we ended up calculating this probability, the time it takes to happen, and it turned out that we could do some analysis, for example, when there was no unbinding. When you have unbinding, it's very hard. We didn't know how to calculate explicitly how it depended on the number of sites, and we had to use simulations. All right, so let's stop here for this mean time to threshold. There is still open problems in terms of the asymptotics. Here it's unpractical. Once you go to this pass and counting pass, it's very hard to get asymptotic formula in closed form, or where you can see the dependency in the parameters. Yet, for the case of plasticity, we are still very much interested in how to calculate the time that a certain number of molecules have reached a threshold, especially when molecules are not completely independent but are organized in a ring. Because you can have molecules like this. This is the next stage we're trying to investigate. That is, they're forming some kind of annulus, and there might be some correlation, some dependency. Once you have one activated, it may impact the probability that the other one being activated. So there is new development in this direction. Okay, but so I wanted to show this because in this program over the last 4 or 5 months, we've seen a lot in terms of simulations, but in terms of analysis of time and getting asymptotic expression, the methods are really different from just the Gillespie algorithm. You have really to attend to the analysis of Markov chains. And what was important in all of these cases is to put the right boundary conditions, and then the rest is not very hard. It's routine